Hello and welcome to episode 5 of the Bio 110 Recap Podcast. We are here to help you digest and, let's be honest, survive some of the essential but complex biology topics you're tackling this semester. Today we've got a jam-packed episode as we dive deeper into cellular borders and the mechanics that keep our bodies running smoothly.
Yes. This week in class, we started peeling back the layers of the cell, literally. We've got so many fascinating mechanisms to discuss, like the semi-permeability of cell membranes and how passive transport is honestly just like going down a slide. You're going to love this stuff.
And for those of you who might have zoned out during lectures or just need this content repackaged, we've got you covered. Now let's jump right in and make sense of this critical biological system.
Let's do it.
All right, so as we dive into the cellular borders we were just talking about, let's start with the cell plasma membrane. It's far more than just a border. It's like the ultimate security checkpoint for our cells. Imagine a line of folks outside a high-end club. Only some people get in and the bouncer's seriously picky. That's exactly what the plasma membrane does.
Okay, I like where this is going. So who, or maybe what, is playing the role of the bouncers here?
Great question. First of all, the plasma membrane is made up of this incredible double layer of phospholipids. It's called a phospholipid bilayer. These aren't just random blobs of fat, though. They're actually highly specialized. You've got these hydrophilic or water-loving heads on the outside and hydrophobic, water-hating tails on the inside, creating this amazing kind of sandwich that blocks, or allows, access.
Wait, and this layer doesn't just fall apart?
No, it doesn't, thanks to some brilliant molecular engineering. That's where cholesterol comes in. It's like the duct tape that holds everything together, balancing flexibility and structure. If the cell gets too warm, it prevents it from becoming too floppy, and if it's freezing, it keeps things from locking up. Genius, right?
Totally. So it's adaptable, kind of like temperature control for the cell. But there's more to it, isn't there?
Absolutely. You've got proteins embedded throughout the membrane. Some stick out and others tunnel through it. These proteins do all sorts of cool things, like controlling what comes in and what goes out, signaling between cells, or even identifying each cell as a trusted team member. One of my favorites is the glycoprotein. It's basically a little name tag sticking out of the cell surface.
Oh, nice. So it's kind of like a badge saying, I belong here.
Exactly. Without it, your immune system might think your healthy cells are intruders, which can lead to some pretty serious problems like autoimmune diseases.
Got it. So we've got a flexible yet strong membrane with security personnel and name tags. Anything else gluing it all together?
Well, the real beauty lies in its selective permeability. Like I said earlier, it's the ultimate security checkpoint. Only certain molecules get in, and it's incredibly efficient. Think oxygen breezing through while sugar needs special permission via these protein channels.
Fascinating. It's a controlled chaos that somehow works flawlessly.
Exactly, and there's so much more to it that we can't see with just a surface-level glance. But yeah, if you zoomed in on a plasma membrane, you'd see a bustling, interactive world.
So we just talked about how the plasma membrane controls what comes in and out of the cell. Now let's dive into one of the ways this happens. Passive transport. It's like having the wind at your back, no energy required. Molecules move from areas of high concentration to low concentration, a bit like how a crowd naturally spreads out into an open space.
So it's like when you open a window in a stuffy room, and the fresh air just flows in without you having to do anything?
Exactly, that's diffusion. Molecules like oxygen or carbon dioxide travel on their own through the cell membrane, as long as the conditions are right. But for bigger or charged molecules, it's not that simple.
Wait, so they don't get the easy pass? What happens then?
Nope, they need some help. Enter facilitated diffusion. It's still passive, still no energy required, but it uses these incredible proteins embedded in the plasma membrane to act like gateways or tunnels. Picture these proteins like guarded doorways, where molecules like glucose are politely let in if they have the right access code.
And the protein acts as the bouncer, letting the approved guests enter. Got it.
Exactly, it's selective but friendly. Now shifting gears a bit, let's dive into osmosis. Think of it as diffusion's cousin, but specifically for water. Water moves across the membrane to balance concentrations of solutes like salt or sugar on each side.
So it's about balancing the stuff in the water on either side of the membrane. But why does water care so much about balance?
Great question. Imagine you have a pile of salty fries sitting in a soggy paper bag. Ever notice how the water from the bag gets sucked into the fries? That's osmosis in action. The salt on the fries draws water toward it to create equilibrium or balance in concentration. It's the same inside our bodies. Water moves to create balance between the fluids inside and outside our cells.
Whoa, salty fries. It all makes sense now. So what happens if the balance is off?
Oh man, the consequences can be wild. Imagine the environment outside your cells is hypertonic, meaning saltier or more concentrated than inside your cells. Water will rush out of the cells and they'll shrivel up like raisins. That's bad news for cells, especially ones like red blood cells that need their shape to function properly.
What about the opposite? A hypotonic environment.
Exactly. In a hypotonic solution where there's less solute outside the cell, water floods into the cells. Too much water and pop goes the cell. This is why IV solutions aren't just pure water, they're carefully balanced isotonic solutions to keep your cells happy and stable.
That's fascinating and makes me appreciate how much our bodies regulate things. And I guess plant cells have some tricks up their sleeve to deal with water movement?
Oh, totally. Thanks to their rigid cell walls, plants love hypotonic environments. Water rushes in, inflates the cell, and creates a nice firm structure. But if you give a plant salt water, yikes, you'll see the cells shrivel and the plant wilt. That's plasmolysis in action, and why salt water and plants don't mix well.
Got it. So passive transport is all about letting water and molecules follow the rules of physics, and it's surprisingly efficient. Anything else?
That's the beauty of it. It's simple and elegant. With diffusion, facilitated diffusion, and osmosis working together, our cells can maintain just the right balance. Nature really nailed it with this one.
Passive transport is such an elegant solution. Letting molecules in water follow their natural paths is amazing. But now I'm curious, what happens when a cell has to go against that flow? Like if it needs to push molecules where they don't naturally want to go.
Oh, now we're talking. Active transport is like swimming upstream. It takes energy to push things where they wouldn't naturally go. This energy comes in the form of ATP, or adenosine triphosphate, which I like to call the cell's molecular fuel. It's what powers everything from muscle movement to maintaining these concentration gradients inside cells.
ATP keeps coming up. It really seems like the VIP of biology.
Absolutely. ATP stores energy in those chemical bonds, and when you break one, it releases that energy to do work. Like moving molecules from an area of low concentration to high concentration, you're essentially forcing these molecules into crowded spaces.
That's some serious determination. So how does the cell actually accomplish this? Are there specialized structures involved?
Oh, yes, and here's where it gets really cool. One of the stars of active transport is the proton pump. This incredible protein sits in the cell membrane and uses ATP to move protons, or hydrogen ions, from inside the cell to the outside. It's like loading up water behind a dam. You create a buildup of positive charges on the outside, and that creates an electrochemical gradient.
Okay, so it's like setting up a pressure system. And the gradient, what exactly does that do?
The gradient is where the magic happens. It's not just about concentration anymore. Because we've separated charges, we've essentially turned the cell membrane into a tiny battery. It's this stored energy that powers so much of what our cells need to do. And with co-transport mechanisms, it gets even cooler. You pair the flow of protons back down the gradient with the transport of something else, like sugars. It's like letting water flow through a dam to generate electricity, which then powers machines.
That's such a great analogy. You're harvesting energy from one process to fuel another. What's an example of this in action?
Oh, there are tons. The H plus sucrose cotransporter is a classic one. As protons flow back into the cell, they carry sucrose along for the ride. It's efficient, creative, and honestly, downright brilliant. We're talking about a system so elegant that even engineers draw inspiration from it.
It's incredible how much is happening at such a microscopic scale. All these pumps and gradients are hard at work, even when we're just sitting here.
Exactly, it's a full-on symphony. And without these active transport systems, our cells wouldn't get the nutrients they need or be able to expel waste properly. They're essential for life as we know it.
Well, that's the perfect note to wrap things up on. This episode has been packed with insights, from passive diffusion to these powerhouse active transport systems. It's mind-blowing what our cells manage to accomplish every second of every day.
Totally. Once you understand how these systems work, you start to see the beauty and brilliance behind the science. Thanks for exploring it with me.
Of course, and to all our listeners, thank you for joining us. We hope this made the intricate world of cell transport just a bit clearer and a lot more fascinating. Until next time, take care and keep learning.